Hey guys, welcome back. We are going to discuss electrophoresis. This is a method to separate macromolecules within an electric field. It is used for nucleic acids and for proteins. And for proteins, we use a special polyacrylamide gel as a support media, and we use sodium decyl sulfate to actually denature the proteins. So this is called a denaturing gel electrophoresis. There is a method that does not denature proteins, so they stay in their tertiary and quaternary secondary structural forms. This was the system most people use was invented by a guy named Lamley, and he was the first to detail studying proteins with this polyacrylamide electrophoresis. So it's usually called STS Page polyacrylamide gel electrophoresis is what Page is stands for. So we have gotten to this point where we've purified proteins from fractionation. And so now we've got fractions that have potentially one or more proteins, probably more than one. And we need to then further purify, or we may have a fraction with just one. Electrophoresis gives us the opportunity to visualize the amount of protein, the molecular weight of protein, and there's lots of applications after a protein sample has been put through an electrophoresis protocol. So it's a continuation going down from where we started with cell lines. We broke apart the organelle. Well, first we classified by the flow cytometry and FACS, and then we were down to the organelles, and then we fractionated from organelle samples, and now we've got samples that contain multiple proteins, and so it's running an electrical field. It's moving of particles that have to be charged. So part of this process, the material we're electrophoresing has to have a charge to it. Cat ions move to the cathode, and ions move to the anode. This technique separates proteins. Let's just say they're also nucleic acids, but for right now, proteins by their different rates of travel. So you could imagine different size proteins, etc., moving differently. And so usually it's proteins. Proteins could be chewed up by proteases. We could look at peptides or nucleic acids, DNA or RNA. Okay, so you have an opportunity to separate lots of different macromolecules. So that's called electrophoresis. So this is kind of the overall where we're going. This is the apparatus that is used. This kind of jello looking thin slab is called a slab gel, actually, and there are these little wells where sample is applied, and then there is a buffer, kind of the upper and lower chamber, and notice there's negative and positive charges here, so there's an electrical current moving down, and the direction of electrophoresis moves in this direction. Okay, and so what happens is the mixture of molecules, notice as we apply electrical current that the different size proteins separate. And it's a lot like gel exclusion, except we're using an electrical field and we've charged these proteins. The gel fractionation or size exclusion was using different pores, and these little snake structures represent different porous molecule, the polyacrylamide that we constructed here. And by the way, you can control the amount of pores in this gel depending on the chemical makeup of this slab. So that's kind of the overview as we separate our samples from the fractionation and apply electrical current and separate the proteins further. And so we have two different types of electrophoresis. One is agarose electrophoresis. That is for DNA and RNA. Okay, and it's a highly purified polysaccharides from seaweed eventually, or from agar, which is from seaweed. And the pores are really large and it doesn't work well. This is in figure A showing the pores. Okay, too big. The pores are too big. For protein work, notice the pores are very, very small, okay? And we can change that by the chemical formula we make these slab gels. It's known as poly, so it's multi-acrylamide. So there's an acrylamide submonomer that we link together, so it's called polyacrylamide gel, and it's held together by cross-linking chemical structures called bisacrylamide. And we make a structure kind of like this and apply a current charge proteins through. Kind of looks like this. If you think of numerous pancakes that have this kind of sieving structure, these are the blue is the acrylamide monomers, and the red is the bisacrylamide. So if you think of these as one direction on your fence, this would be the cross-linking between the two chains in a fence here. So we have cross-linking here, cross-linking there. And we end up getting this sieve structure, and there's numerous, depending on how long the slab is, you have a numerous slices of this polyacrylamide here. I think this is kind of colorful here. So this is known as polyacrylamide gel electrophoresis page. There's the acrylamide monomer, and N means multiple numbers. And there's the acrylamide structure. And you get the cross-linking with bisacrylamide. They are much tougher than agarose gels. You could drop a polyacrylamide gel on the floor and it will pretty much stay intact, whereas agarose will break apart. Okay, polyacrylamide is thermal stable, transparent, strong, and inert. Those are really key components here if we're going to separate proteins. The gels have no charge, and you can control the pore sizes. And so we end up separating proteins based of charge to mass ratio and molecular size. Okay, and that's also known as molecular sieving. Sieving is purification. I don't know if you know about cheesecloth, but it's a method to make cheese, and you get the whey and other materials out of the cheese curds, and you bind the cheese curds together to make a block of cheese. Well, this is a lot like that sieving mechanism, a filtering mechanism. They're very stable under temperature because you apply electricity, and that's going to heat things up. And a different range of pH, again, different pore sizes, and the separation speed is very good. So again, here's another cartoon showing the acrylamide, and they're linked together by bisacrylamide. Here's its structure here. So you need to make that, it's the chemical reaction, use ammonium persulfate and a chemical known as T-med. So you mix kind of a brew of buffer with certain electrolytes in it and the monoacrylamide and bisacrylamide, and you mix that together and you pour it into a mold. That's the slab gel here, and that's how you and you let it polymerize, and then it makes a complete page gel. This is a cartoon showing the acrylamide monomers and the crosslinks. So there they are, the bisacrylamide. And so you can change the amount of the bisacrylamide and change the number of crosslinks. You see that's a pretty big pour here, but we could make more links if we wanted to and make the sieve size much smaller. Usually acrylamide goes from about 3% to 20%. And so depending on the amount of acrylamide depends on what you're separating. We'll talk about that later. So that's the gel side, and the SDS in SDS page is from the detergent. It's an anionic detergent, means when it's dissolved, they will have a net charge in a wide pH range. And so a protein is really a polypeptide chain. And what happens is the longer the chain, the more detergent it will pick up. Okay, so the bigger the protein, the more negative charge. Okay, and you use the SDS to coat the protein and make a bigger, uh, uh, make the whole protein swamped out negative. So I know we had talked about R groups, and I know we talked about plus or minus amino acids. This detergent swamps everything. It covers all the primary R group charges. It makes everything negative. So therefore, the protein is separated by its size. Okay. The gel will restrain. Okay. That means they will keep large molecules from moving as fast as small molecules. And the mass charge ratio is about the same. And the separation is on the different molecular sizes of the proteins here. And the distance that they migrate in the slab is known as RF, and it's proportional to the log of the mass. So there's actually a mathematical formula where you can predict the size of a protein relative to how it moved in a slab. And so usually we have positive controls right here, if proteins of known mass. So you can order a collection of proteins from different companies called standard masses, and we run those, and those end up looking like bands, and we could calculate the RF. And we know that a certain distance from the well means a certain size protein. So we can plot that as a standard curve and then compare, say, an unknown, maybe it's between these two known, and we could measure this distance and calculate the size of the protein in kilodaltons, DA. Okay, kilo is 1,000 daltons. Remember, one amino acid equals 330 daltons. So if we know its size, we can then divide by 330 and determine the amount of amino acids. And if we know the amino acids, then if we multiply that by 3, we would know the gene size, assuming no splicing, which pretty much you can assume, but you'll know the minimum size the gene could be. So that's how you can link proteins and their genes together. All we have to do is compare standards to unknowns. Here's the chemical structure of SDS. And there's another compound known as BME. It smells, if you know anything about Capto, it's sulfury smell. Smells kind of bad. It's also known as BME. BME. Okay, ME if you see BME, that's beta-mercaptoethanol and SDS. And so here is the SDS. It's a detergent that denatures, so it breaks apart secondary and non-disulfide linked tertiary structures. So it breaks apart the protein and makes them primary down to their primary sequence, and it applies a negative charge to each protein. So here's a protein, it's smaller than B, A, and B. And if you counted the number of charges, B would have more charges. Okay, so we take a protein, say it's a dimer with two subunits, A and B, and they're held together by a disulfide bond here, bridge. We can use BME or DDT, which will break apart these two proteins. You add this detergent, and now we separate. Here's B and here's A. So SDS applies a negative charge proportional to its mass. Big proteins are more negative, they move slower. Small proteins less negative, they go through the sieve quicker. Okay, so this is the theory. This one slide is the theory of how an SDS works. So if you were having to describe that, you would certainly include all these elements here to discuss SDS page. And again, the mobility, the RF of a molecule is determined by its, if it was in a free solution, okay, with zero gel, zero, and then the gel matrix. So it's a comparison here. So what happens is the Y0 is the same for all species, and then that means the mobilities are determined by the sieving action, and the sieving action is determined by the molecular weight, okay, equaling the charge to mass ratio of all proteins. It's like large and small proteins here. So if we look at log molecular weight and plot that versus the RF of known proteins, it pretty much works for all proteins except those that are extremely large or sometimes small. Notice we get out of the linear range. Okay, so this is good for proteins in that range. Just be aware if you're dealing with extreme large or extreme small, you may miscalculate the molecular weight. Here's another cartoon showing the same thing, that the proteins are broken apart. And here's A and B and C. They have a charge and they separate out according to their mass. Okay, according to their mass. Here's C, here's A and B. And we'll talk about how do you visualize proteins that have no color and yet they appear blue. Well, there's a dye called bromophenol blue, and there's a dye called, which is a tracking gel, and then a tracking dye, and then there's Kamasi blue, CB, Kamasi blue, which stains proteins, and we can visualize them because this slab is clear. It's like cellophane, it's just clear. Okay, and if you don't stain the proteins, you don't know where they're at. So this is a cartoon showing the process here. This is an extreme cartoon, and this is from Wikipedia, so you could look at this. But long story short, they take you step by step through the protocol here, and they're showing you weigh out the tris, the SDS, the acrylamide, the bisacrylamide. You take fairly pure water, a little bit of acid here, you adjust the pH, you take out the extra gases under vacuum, and then you add the catalyst, ammonium persulfate in TMed, and then you end up pouring, stacking, and separating gels. We'll talk about that. And so here is a separating gel. That's the main slab right here. And then on top of that, you add a stacking gel. So this is a discontinuous structure here of a slab gel and a stacking gel. So it's actually two different acrylamide gels in one. And we'll talk about the reason you actually need that in separating proteins. But for now, and this takes a couple hours to polymerize. Okay. So we're back here, and okay, now we're back. Okay, trying to figure this out. Don't know. So we left off with staining and getting proteins separated here. And again, this is a look at the slab, and we have a stacking gel and a so-called separating or running gel. Here's the sample wells. The sample again has a glycerol sucrose. It's heavier than water, and it has a dye, so you can actually see it. And if you can tell, this is an open chamber right here. It's full of the top buffer, and this is the lower buffer here. Okay, and again, this has no any kind of this is held together. It's actually hard to see, but it's a spacer, and basically load it. I'm sorry, and big proteins will move slower than small proteins, and this is the direction. Glass, and these are glass plates as part of, and acrylamide does not stick to glass, and there's a spacer piece of plastic, and this is just you apply electrical current. Takes a couple hours to separate. And you're saying, well, how do you know where you're at? Bromophenol blue runs at the dye front, and so you run it till the bromophenol blue is at the bottom, and then you have you can separate the proteins. So what's this purpose? Well, the stacking gel, the upper gel, is needed to concentrate all the proteins in the one band. So it's the same concept as a starting gate. Okay, you put all the horses in the starting gate. Some horses are bigger than others, and you put them all together and they start at the same position. So you apply electrical current, and all the proteins stick together, and they hit the separating gel and they slow down, and then you turn on a different, usually this is slow electricity, and this is, you have a lot more power here. So you line them up, and remember that's bromophenol blue right there. So BB, that's bromophenol blue, and they all line up at the beginning, and you apply your current and you separate based on mass, large and small proteins here. So it's a discontinuous gel. In other words, this is one type of acrylamide linkage pore size, and it's very has a lot of space, and this is a very packed, smaller pores. So we allow everything to stack up, and then we apply a current to separate them out based on that. And it's about a 3% acrylamide versus a 7.5% acrylamide. So big, they call it resolving gel, separating gel, gel slab, whatever it's called. And the buffer has glycine, chloride in it, and those carry. And remember, the leading dye is following these chloride ions in the upper chambers buffer here, and that's bringing the proteins, and they hit this and they slow down, and then we separate the proteins by molecular mass. So and this is known as vertical gel electrophoresis page, and it's a double layer, and we've been talking about SDS. When you don't use SDS, that's said to be native electrophoresis, and that's why you maintain proteins in their natural conformation. And so sometimes you may want to do that for purposes that we're not talking about now, but I just did want you to know that we can do native gels as well as denaturing gels. So these proteins are stretched out to the primary, and these maintain secondary, tertiary, and sometimes even quaternary structure. Proteins are amphoteric. They have R groups that are positive, negative, or zero, and depending on their pH. When a protein charge is zero, it's said to have reached its isoelectric point. This is where you've changed the pH such that the R groups, positive, negative, or actually have a net charge of zero. And every protein has, because it has different R groups, different amino acids, has a PI, a point, and there's software, the bioinformatics will allow you to separate proteins based on their electric point. And when we talk about two-dimension, page, SDS page, okay, SDS, okay, page, I didn't spell P right, okay, should be a P, and that's a P, so whoa, so 2D electro SDS page here, and we'll talk about isoelectric electric point. All proteins have a different one unless they're very closely related, then they'll be close together. This just shows the blue dye here, and it's heavier than water, and this is the power supply. We have the tracking dye, bromophenol blue. It's usually a glycerol or sucrose. And this is the stacking gel, has a different percentage acrylamide than a running gel, 5%, 12. The proteins still have to be heated. That heat applies the detergent carefully and breaks apart with DDT, and BME breaks apart the proteins to the primary structure. You load it, so you have the tracking dye and the glycerol, and you apply voltage 20, 30, 40 minutes, maybe a little longer, depends on the size of the gel. And then you stain and you image the gel. So every protein, you stain it, that's data, and that data can be put in a database. So can you imagine if we were looking at, say, a disease cell line and normal, so cancer, non-cancer, and we looked at proteins, and you might see different proteins? That could give you an idea of what proteins are involved in the disease. Okay, so just imagine the example. So after the slab gel is run and the tracking dyes made it to the bottom, then we take the gel, we break apart the spacers and the plastic plates carefully, kind of pop it, and the acrylamide doesn't stick to the glass. The glass is smooth. You can also apply a silicone, kind of like kind of a non-stick Teflon, and that will allow the glass plates to separate. But anyway, you get the slab, put it into a container with methanol, acetic acid, and 0.1% Kamasi blue, and you incubate it, and then you destain, and you can visualize the proteins. So these are showing the actual stained proteins here. This is rotavirus proteins. So in the rotavirus, they purified by electrophoresis, but probably purified by some type of centrifugation, and they've got some major proteins in the virus to study. So now, if you have this pattern, you may have a rotavirus in the patient's sample here. You could look at that. And here's stain gels. You can actually dry them down on plastic and preserve them. You can shine a light through them and take a picture, and that's an image for a scientific paper. Again, you can measure the RF if you have standards, right? And we can figure out what these unknown sizes are. Okay. And here's the markers in lane 1. So all these in lane 1 are known molecular weight proteins. You calculate the RF, and then you look at the proteins you're interested in, and you can figure out what they are. And here's showing like myosin's running there, actin, tropomyosin, myosin light and heavy, and here's myosin light chain as well. And you can actually visualize these. Okay, so there it is. This is Kamasi. These proteins have been stained with silver stain and applied to a piece of x-ray film, and it's kind of like the process of photography there, and it's a lot more sensitive, and you can see a lot more finer bands here, so you can look at samples that way. Applications for STS page, 15 or more. You can do peptide mapping. We'll talk about that. We take a protein, we break it chew it by proteases, and we get a so-called fingerprint. And so now we can map any protein with a certain series of proteases and get fragments, and that's the protein's fingerprint. And you can actually, with a razor blade, cut those bands out of electrophoresis, or you can electrophorese out of the bands. There's ways to do that. And you can then actually have a pure protein out of the gel, and you can then cut them and run the fragments. Here's some common proteases that people use, and this is where they cut. Any ARG and lyse are cut by trypsin, and here are some amino acid cuts by papain, chymotrypsin, and some of the other ones there. Okay, I'm not going to go through all of them. So here is another application, and we're going to talk a little bit more about this in just a little bit, probably next series of lectures. Edmond degradation. We can here's known standards. We can actually take each one of these main bands and apply Edmond degradation and get the amino acid sequence from the N-terminus, So these are the amino acid sequences of the N-terminus of some of these proteins. This is a flow chart, N-terminal Edmond sequencing, and it looks like they're looking at some different antibodies here that they were separating here. So that's my 10-cent tour of electrophoresis. Come back and we'll talk about extra fun stuff in the next series, two-dimensional gel. And what we just talked about was one dimension. Okay, we're going to change the vertical and the horizontal with two-dimensional gel electrophoresis. Okay.